The overall goal of this procedure is to incorporate GAG genes derived from subtype C HIV-1 infected individuals into a replication-competent HIV-1 plasmid in order to assess the effect of GAG sequence polymorphisms on HIV-1 replication capacity in vitro. This is accomplished by first amplifying the GAG gene from the patient's plasma. The second step is to clone the patient-derived GAG sequence into the replication-competent infectious molecular clone MJ4. Next, the GAG MJ4 chimera plasmid is transfected in order to generate infectious viral stocks and titered to assess infectivity on an indicator cell line. The final step is to measure in vitro replication capacity in the GXR25 T-cell line. Ultimately, a radio-labeled reverse transcriptase assay is used to quantify virion production over time in order to visualize viral replication kinetics.
The method we're going to describe today can help answer key questions in the HIV field. These include defining the determinants of HIV replicative fitness and understanding how HIV fitness affects long-term pathogenesis. Describing the methods today will be Jessica Prince and Dan Claiborne, two of my graduate students.
To begin this procedure, reverse transcribe.